Hey, Carlos, long time no see.
Yeah, yeah. Hi, Greg.
How are ya?
Still look the same.
So do you, man. Younger than ever.
Yeah, sure.
Great. They've decided to mow right outside my window right now, of course. So.
Murphy.
Yeah.
And I see Tom too. Good to see you, Tom.
Hello, Carlos.
Cesar, mucho gusto.
Gracias. What is see Dr. Isaac is here, Dr. Soar, and just waiting for Dr. Cairns, I guess.
He's just joined, so we're all good. There he is.
I'm here.
Okay.
Still, we'll wait till 11:01, basically give people
Yes.
a few extra seconds, and then we'll get started.
Mhm.
All right, you got people tuning in from all over the world here. Very nice.
2 continents, at least. No, 3. I'm expecting a couple from China.
Ah, okay. I was trying to figure out what the third one was, but yeah, that makes sense.
I see you're in your office, huh?
Yeah, they're mowing the grass right outside, but I think he just did the closest pass to my window right now, so it should be good. I'll mute my microphone once we get started anyways.
Yeah.
I can't hear anything.
Ah, good. Okay. All right, last few seconds for people to go ahead and join us. All right, we'll go ahead and start. So I'd like to welcome everyone to the Master's Defense of Cesar Valencia y bienvenidos familiares y amigos de Cesar también.
Gracias.
Thanks. That's the best I'm going to be able to do with Spanish. So the apologies for that. So Cesar is defending his master's thesis today, but he's actually been in the lab here for 11 years now. He was the very first person that I hired when I first started, and we both started out in an empty lab that had zero equipment. So he has been here from the ground up. He's an absolutely critical member of the team and has been for all of the students and personnel and visiting scholars that have worked in our lab over the past 11 years. Sometimes I try to sneak slides into presentations, but it turns out Cesar at the beginning of his presentation is going to show you a defining moment of his time in the lab. So I'm just going to let him show the good pictures. But of course I have a couple good stories. So for instance, when we first hired Cesar, I didn't have any cotton experience whatsoever, really. And Cesar came in and said, I used to manage a several thousand acre experimental cotton farm in Peru. And I said, this interview's over, you're hired. And that was it. We took off from there. The first summer we were out in the field doing field trials. We're up to our knees in mud, putting out PVC pipe to irrigate. And Cesar's like, man, in Texas, you guys grow cotton like it's a third world country, right? And so later, a little later in the year, we're up in Lubbock doing some field work. And all of a sudden we hear pop, 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 gunshots going off right nearby us. And so the next thing you know, we're laying down, hiding beneath cotton, trying to figure out what's going on. And we're pretty sure the person wasn't shooting at us, we were shooting at something else nearby, but we decided we would just pack up and not sample that field anyways. It wasn't that important, so we got out of there. And then much more recently, for those of you who are familiar with the ice storm and the pipes that broke in the ceiling and flooded the lab, Cesar and I were in here trying to save equipment and samples and things, and we're in ankle deep water, and water's raining down from the ceiling and stuff's collapsing all around. And Cesar's like, man, I thought things were going to be different in America. And I said, well, no, welcome to Texas. So we're all just sort of one step ahead of the apocalypse here. So despite all that, Cesar has stuck with the lab, and he already had a master's degree to start with in agronomy. And he decided he wanted to get a master's in entomology. So we fully supported him in that. And in addition to doing his own research, of course, as lab manager, he's been invaluable in helping out others with his research. And he's developed a pretty amazing and broad scientific toolkit that he can now draw from. And you're going to see a lot of that on display here during his master's presentation. So everything from microbiology, he wanted to not perfect, but to expand his microscopy skills. So we enabled him to do that. And he also wanted to start learning some molecular biology. So you're going to see him reporting on some gene expression assays. So it's all really impressive. So the real danger in all this, though, is Cesar's going to progress so much and go on, and he's going to overqualify himself to be my lab manager, and I won't be able to afford him anymore. So that'll be a good success story for Cesar, but unfortunate for us in the long run. Okay, so I'm gonna pass it over to Cesar, and if I will mute my mic. It looks like everybody else is muted, but just in case there's any noise, let's make sure our mics are muted. And Cesar, take it away.
Thanks, Dr. Soor. I'm really grateful to be part of your team. And hello, everyone. Thank you for joining me today. Hola, amigos y familiares. Gracias por acompañarme. That's the old Spanish I'm going to speak. And with that, I'm going to share my screen. Let me share screen. It's going to be. See. This thing. Oh, I quit. No, I'm here. Okay, yeah, see now. Okay. Okay, do you see my screen?
Yes.
Okay. So today I'm gonna present my thesis work that was titled An Integrative Analysis of Cotton Fungal Aphid Interactions. The committee members are Dr. Gregory Soar, Dr. David Kearns, and Dr. Thomas Isaac. So I'm gonna, as a icebreaker, I'm gonna tell a little story about my experience as a student here at A&M. So this photo here was taken during my first day of classes. That was back in 2018 when I started this master's. Before this day, the last day I was in school was back in the year 2000, when I was back in the day when I was still living in Peru. It's been a really great life experience for me here at A&M. And talking about life sometimes gives you some hints or signs about the future. This is the one I got during my second date as a student. And I interpret this sign as it won't be easy, but it will be worth it. Is how I survived this thing without scratches is one of life's mysteries, because it was really bad. Anyways, so that was it about my life as a student. Now with the real business. So my thesis is structured in 3 chapters. I have chapter number 1 is just the classical introduction. Chapter number 2 is about the interaction between the cotton plant, the aphids, and the fungal and the fungi that I use. In chapter 3, we wanted to, it's kind of like a follow-up of this thing. We wanted to see what happens with the spores after I sprayed the plants. So it was titled Microscopic Observations of Fungal Spores on Leaves, 2 genotypes of cotton. We're gonna start with the introduction. So I use cotton plants. The cotton plant is a major global agricultural crop. Is China, India, and the US are the 3 largest cotton producers in the world. It's grown mainly for its fiber, but it has other uses. Animal feed is one of them. It's a major cash crop in Texas. This year it's been reported that 6.5 million acres were planted. And about 90% of the cotton planted in the US is upland cotton and is genetically modified. The 3 major pedigrees planted are Delta Pine, Americot, and Phytogen. The cotton aphid is the insect I use in my study. Its Latin name is the Aphis gossypi. It's a flowing sucking insect. It's well known in cotton production areas. It's a secondary pest, usually. Sometimes it could be a problem, especially at the end of the season when we have the fiber already opening the bolls. Could be more serious there, but usually it's just secondary pest. It reproduces asexually. It also goes on sexual cycle, but it's more commonly as a sexual reproduction. Life cycle, it takes like a month. And very important, so aphids are not always a bad thing in the field because many generalist predators feed on them. I guess that's why one of the reasons, maybe in the US it's not that important, but in countries like in Peru where we don't use GM cotton varieties, having natural enemies in the field is really important. Okay, so plant fungal endophytes. I'm gonna introduce this concept because it's very important it was very important in my research. So fungal endophytes are defined as species of fungi that can live within plant tissues with producing no symptoms. The fungus, these species are able to colonize, and you are not going to notice any signs of this thing unless you isolate these species. And the way I did here is, in this case, is Bouveria bassiana growing out of cotton leaf tissues. And that's the way fungal endophytes are recovered from plant tissues. Continuing with fungi, in this study, I used 3 species of fungi. One of them is a well-known entomopathogen, Bouveria bassiana. I also have Chaetomium globosum and Phyllalemonium inflatum, which was known as Bacillomyces inflatum a few years ago. So Bouveria bassiana was obtained from Botanic Garden, which is a commercial product, and Chaetomium globosum and Phyllalemonium inflatum were isolated from cotton in Texas. A little bit of the fungal species. So Bouveria bassiana, as I said, as I mentioned, is a well-known entomopathogen. It is available commercially, normally for foliar treatments. It's a soil-borne fungus, and it's been reported as an endophytic fungus in several plant species. And in this photo that I took here, you can see the reproductive structure of the fungus, Bouveria bassiana. Its spores are about 5 microns diameter, rounded. The next one is Phyllalemonium inflatum. Was isolated from cotton squares here in Texas. It's reported as an endophyte in cotton end peppers, usually referred to as a compost-dwelling fungus. In the photo here, you see the reproductive structure of this species, Phyllalemonium inflatum, is completely different as Bouveria bassiana. This one produces like a long chains of spores. And the spores are usually like a lemon shape and about 3-4 microns in size. The third one is Chaetomium globosum. Chaetomium was isolated from cotton leaves here in Texas. Is being reported as an endophyte in different plant species, and is usually referred to as a saprophytic fungus. This species of fungi reproduces through producing fruiting bodies that are perithecium, and the conidia are, conidia are inside these fruiting bodies. Conidia, they resemble American footballs, and they're about 10 microns. Now, I'm gonna talk about a little about the plant defense system. So plants have a really complicated defense system to fight against biotic and abiotic stimuli. And I'm gonna focus on the induced resistance because it's more important for my research. But plants also have this constitutive resistance, which is something that the plant produces continuously. Is for example cell walls, trichomes, waxy cuticles, or even some compounds like gossypol, for example. Gossypol, the cotton plants produce gossypol all the time. It doesn't have to be induced. The plant produces it always. And before I go into this into this induced resistance, I'm gonna talk about a little about some important concepts that we need to remember when it comes to resistance. One of them is the MUMP, which is the microbe-associated molecular patterns. These MUMPs are important for the plant recognition. One of these important MUMPs is the fungal Kaitin. Fungal Kaitin is a very important compound of the fungal cell walls, and is a well-known conserved MUMP. Another concept is the MUMP trigger immunity. So when the plant recognizes these MUMPs, the plant triggers the defense system, and it does this based on some receptors that are located on the surface of the leaf. So the plant, and this is very important because this is the first step in the battle against the pathogens in this case. So the plant must be able to recognize the MUMPs that land on the surface of the leaves, and that is when they start fighting and usually destroying all these enemies. This is why most plants are resistant to the majority of pathogens. Another important concept is priming. So once the plant has detected these pathogens, it has destroyed these things, it kind of memorizes these things. And then next time this thing happens, the plant is able to respond faster and stronger. That's what is called priming, which is a physiological state. And the other one is crosstalk, and it's pretty like self-explanatory. So plants are always, all the time, being exposed to biotic and abiotic stresses. And when this happens, there's several plant hormones that are being triggered in this plant system. So the plants need to coordinate how these salicylic acid, how these jasmonic acid, how those other hormones like gibberellic acid, indole acetic acid, you name it. There are many. So the plant needs to coordinate this thing to better respond to the attack. And in some cases, there is an antagonism in between these plant hormones, meaning so one is going to be down-regulated and the other one is going to be up-regulated based on the plant needs. And maybe that's because some breeders, they look for plants that will produce more, that will grow more, but at the same time they're going to lose their defense activities. And now we're going to talk about this induced resistance. So when it comes to induced resistance, it's been identified that we have two systems. One of them is the systemic acquired resistance, which is based on this hormone here, the salicylic acid, and is being triggered by sucking insects like the aphid here, or biotrophic pathogens. So the signaling molecule, as I said, is salicylic acid, and sometimes the elicitors, in this case, this bacteria here, will produce these things that are called a hypersensitive response. And when this thing happens, the systemic acquired resistance is triggered, and then the signal travels across the plant. That's why it is systemic. So the plant is going to be protected not only here, but also on different parts of the plant that were not under attack. same with the induced systemic resistance. This one is triggered by herbivores like caterpillars, and also by necrotrophic pathogens. Also, we have some elicitors like these bacteria here that work in the roots, and then they stimulate this system. The signaling molecules in this induced systemic resistance are the jasmonic acid and the ethylene. Regarding the crosstalk in this part is well known. It's kind of like a generalized to say that jasmonic acid and salicylic acid are in general antagonistic. Okay, so now we're going with the actual research chapters. Chapter number 2, cotton genotype mediates foliar elicitation of plant defenses by fungal spores and the resulting effects on cotton aphids. We have 3 objectives for this chapter. The first one was to evaluate the response of cotton aphids on cotton plants treated with either viable or dead fungal spores. So now, at this point, maybe some of you will say, why Cesar is using dead spores? So the reason is we wanted to see if the differences, if the response from just spraying this viable spore was because of the presence of these things or something else was happening. Like for example, when you spray these fungal spores, and then you contrast this thing with dead spores. If you have the same response, you could say, oh, it's because it's just the presence of these things is the ones triggering the response. That's why we included dead fungal spores in our study. So then, kind of like a follow-up, we tested the response of cotton aphids on plants previously treated with chemical elicitors and a source of MUMP. In this case was chitosan, which is some sort of chitin. And last but not least, we tested the whether foliar application of spores affect expression of genes in the ISR or SAR defensive pathways. Okay, we're going to start with objective number 1. So in this objective, we just wanted to see what happens what happened with aphids after you spray the plants. This is very important. So I'm not spraying the aphids. I'm using one of these fungi species is an entomopathogen. But in this study, I'm not using these fungi as entomopathogens. I'm using these things to stimulate the plants, and then I use the aphid. I put the aphids on the plant. So very important to remember. And we have 2 species of cotton. The first one is a Delta Pine 0912, and the other one was a Phytogen 367. So Phytogen is a species, is a genotype of cotton that is known to be nematode tolerant. Both of them are resistant to insects because of the Bt, and also resistant to herbicide. And then we have the 3 species of fungi, Chitomium, Philemonium, and Boveria. Then we have the 2 states of fungal spores, viable spores and dead spores. These dead spores were heat killed. We have cotton aphids mixed stages, and then we have pods, organza pouches to prevent aphids from moving across plants, and soil mix. Okay, so this is part of the methods. This is how we produce the spore suspensions. So I grew these 3 species of fungi in nutrient media. Once the cultures are mature, usually after 2 weeks, they have plenty of spores to be harvested. And I do that, but when you harvest from these dishes, you have the hyphae and spores. We need to separate these things, and we separate these the spores from the hyphae using sieves. Then when finally we get the pure spores suspensions, then we proceed to calculate the number of spores per cc, and then adjust to the desired spore concentration. I usually prepare 200 mils of spore suspensions, and from those 200 mils, I separate two batches, 100 and 100, and then one of them is going to be used for the viable spore treatments, and the other one is going to get heat kill to be used as dead spores. And to kill the spores, what I did is I put the beaker with those 100 mils of spore suspension in a water bath, 70 Celsius for a couple hours. After that, I tested viability of the spores. So you see on top is where the spore suspension after killing the fungi, and below is the spore suspensions from the viable spore suspensions. Now, this is the timeline of events. Each segment is about 1 week. So it takes, from planting, it takes 3 weeks for those plants to be ready for the treatments. 3 plants, at this time I have plants that are at the second true leaf stage. That's when I spray the plants, in this case with the fungal spores. And I allow 1 week as an incubation period. So after 1 week, 7 days, I go back to my plants and I get these plants infested with aphids. And we place 2 aphids per plant. After infesting, we cage the plants with these organza pouches, and we allow 2 more weeks for the aphids to reproduce on the plant. At the end of this period, I take the plants and I count the total number of aphids per plant. Now the results. I spend a lot of time counting aphids. So this is the result of the generalized linear model with a negative binomial distribution. Most of this aphid data is always non-parametric, and that's why I use this model. So we have 4 different trials for dead spores and 3 trials for viable spores. In the analysis, you can see there is significant values for in this case for viable spores. The genotype is different. The trials are different. The treatment is not. And we also have a genotype by trial interaction that is significant. Then when we see what happened with dead spores in these 2 genotypes, we have genotype is different, trial different, and then we have treatments is significantly different. So meaning we have something going on here with dead spores, but not here. And then of course we have the genotype and trial interaction that is significant, and genotype by treatment interaction that is also significant. Because of that, I'm gonna show you the graphs separate, and I'm only going to show you the result for phytogen when treated with dead spores, because the other combinations I didn't see anything. So it's just to save time, I'm going to focus on the ones where I saw things. And as I said, there's 4 trials. We have aphid counts on the X, and trials time on the on the Y. Sorry, aphid counts on the Y, and trials time on the X axis. And then you can see that across trial, and you can also see the differences in the aphid density, aphid numbers is different. You know what is different? Is because these things were done at different times of the year. And of course, sometimes it's hotter, you get more aphids, sometimes it's cooler, fewer aphids. But what is important is I noticed significant differences in the number of aphids, but having more aphids than the control. Control is the blue. So in this case, I'm having more aphids significantly for Bassiana and for inflatum. Globosum it has the same trend. In the second one is Bovvaria significant, not the other ones. Then here again, Bovvaria and Globosum. And here again, Globosum. Bovvaria following the same trend, but not significant. And same with the inflatum. Now, I was like this guy. I wasn't really expecting these results. To be honest, at the beginning when we started this thing, I thought I was going to have fewer aphids with the treatments. And I couldn't explain at the beginning, why is that dead spores are having these effects and not the viable ones? And one genotype is responding and not the other one. So that's when we decided, okay, so I'm gonna use elicitors, because if this thing is because of, like a plant defenses being triggered, I'm going to use elicitors to as positive controls. And I will see what happens when you treat plants with elicitors and what happens when you treat the plant with chitin. And then I'm gonna compare to my fungal treatments. Then, hence, objective number 2, to test the response of cotton aphids on plants previously treated with chemical elicitors and AMA. Okay, so the material in methods are exactly the same as in objective number 1. The only difference is instead of using fungal spores, I'm using chemical elicitors and a source of MAMP, in this case, chitosan. Okay, again, the statistical analysis. Once again, we see genotype differences, meaning their phytogen is different from delta pines. Also, treatment difference, and again, the interaction of trial and treatment with genotype being different. That's why I'm showing you separate trials. You see again, the two ones on top are the phytogens. The two ones here are the delta pines. We have aphid counts on the Y's axis and the treatments here. You see the treatment with the methyl jasmonate and Actigar, which is a non-SAR elicitor, end up with these plants end up with more aphids compared to the control ones. And the repeat trial was exactly the same, only for phytogen. Didn't see response from the delta pines, but you see that has the trend to have more aphid. It has a trend to have more aphid, but it's just not significant. But same as with my treatment with the spores, only phytogen is the one that responds to these treatments. Now I say, okay, so is it because of chitin? Is the plant responding to chitin? So I decided to use chitosan, which is basically chitin, and I sprayed these plants with chitin. And again, with chitosan, I mean, again, you see the response exactly the same as what I saw with the phytogen in dead spores, having more aphids on the treatments. It seems like the plants are responding to the presence of chitin. And delta pine is not responding. Really interesting. Now, are the fungal spores changing the defense gene expression profile? That's when Dr. Shore told me, now you need to start learning molecular biology techniques. I said, well, I'm gonna try. And at that moment, I felt like this guy. I feel like really smart when doing molecular biology. Okay, so we decided to test if the foliar application for spores are affecting the expression of genes in the ISR and SAR defensive pathways. Material and methods. So we use the plants in objectives number 1 and 2. We took samples from these plants at 7 days after treatment. 7 days after treatment, these plants were, I took the samples from these plants. And we use these primers, LOX1 and AOS as jasmonic acid responsive genes, and beta-1,3 glucanase as a salicylic acid responsive genes. I also use actin as the housekeeping gene. The methods. So as I said, I took 100 milligrams of tissue from these plants. By the way, I didn't use the plants from which I took the tissue. I didn't use those plants for the Aphid trials. So after taking the samples for this objective, I just discarded these plants. So we take 100 milligrams, put them in a tube, and straight to the liquid nitrogen, and kept it at -80 Celsius until needed for the actual RNA extraction. So when the time came, I did the RNA extraction using this Sigma Aldrich kit. It takes a lot of time, pipetting and centrifuging. And when you get the good concentrations of RNA, then you go to this machine here, which is a thermocycler, to do the conversion of RNA to cDNA. So once this process has been completed, we take samples of this cDNA and mix with other things like the cyber green, the primers, and a dye to do the final qPCR. The final qPCR, yes, using again this thermocycler. After that, you get your data that is going to be analyzed and using Excel, and you finally get your gene expression data in folds, number of folds changes compared to the control. So here in these two graphs, we see the expression of the AOS and LOX1 genes, which are jasmonic acid genes. And we notice again the gene expression changed only for the phytogen genotype. Here we see the box plots. The red ones are the dead spore treatments, and the green ones are the viable spore treatments. You see this pattern. You have control in the fungal treatments. You see this pattern when the spores are viable. You see this pattern of down regulation that is significant for globosum and inflatum, and a pattern of up regulation when you use dead spores. So it's completely opposite. And with the LOX1 gene, we see the same pattern of down regulation when you use viable spores, and we didn't notice any changes when with the dead spores for this gene. Then when we see to the beta-1,3 glucanase gene, which is the SAR gene, we see the only phytogen is significant, but down-regulating this gene. Now, Dr. Shore said, but we see what happens with the plants after you treat them with the spores. Now we need to see what happened when you treat the plants with the chemical elicitors. I said, okay. So I went back and do treatments, and again do all the process of gene expression, and we see you have here again the full number of fold changes and gene expression. when we treat the plants with methyl jasmonate in delta pines, you see the delta pines is responding to the chemical elicitor. And you have this AOS and LOX1 gene being upregulated significantly, not the beta 1,3, and it's not supposed to. And when you use Octigar, you have a downregulation of the AOS gene and a significant downregulation of the beta 1,3. And this thing is, I don't know why this happened, because supposed to have this thing upregulated, but it's maybe that's one of the reasons why delta pine is not responding. But when you see a phytogen, you see upregulation of the jasmonic acid genes, in this case LOX1. AOS is not different, but it follows the same trend. Ain't nothing going on with the beta 1,3. As I said, it was supposed to be like that. And we have something that surprised me is that when you treat the plants with Actigar, which is a well-known SAR elicitor, of course you have the SAR gene, the beta 1,3 upregulated, but also the jasmonic acid genes were upregulated significantly. You see the AOS and the LOX1 gene upregulated. Maybe this is why the treatments with Actigar, they behave same as jasmonic acid, because it's having all the same effects. Okay, conclusions. So fungal spore foliar applications affect cotton defense responses. Major effects of both genotype and fungal status, viable versus dead. Cotton aphid response on delta pines was not affected by any of the fungal or elicitor treatments. Cotton aphid increase on phytogen plants pre-treated with dead spores, with methyl jasmonate, with Actigar, and with chitosan. And finally, the defensive gene expression was downregulated generally when the spores were viable and upregulated when the spores were dead. And that is the end of chapter number 2. Now going ahead with chapter number 3. As I said, this is kind of like a follow-up. And mostly because I love working with microscopes. We see here we only have one objective, to describe the fate of fungal spores on leaf tissue following foliar treatments using microscopy. So we wanted to see what happened to the spores after I sprayed this on the plants. Are they going to germinate? Are they going to colonize the plant? What is that they do? We wanted to know what happens with this. And then if they germinate, is it possible that we can get a microcycle conidation of these fungi, especially Bassiana and Inflatum? Microcycle conidation is a process of some fungi species, once they germinate on the surface of the leaf, in this case, and they somehow realize that nutrients are not enough to grow vegetatively, then they start the germination process. Then you have the germination tube, and immediately they go into reproductions and produce more spores. This is known as the microcycle conidation. Now we see materializing methods for this chapter is, again, I'm using the same 2 cotton varieties, the same 3 species of fungi, and I'm using this technique, which is called the trypan blue, the lactophenol stain, which uses acetic acid, ethanol, and glycerol. I use clear plastic 6 well plates and microscopes. And I use 2 kinds of microscopes. I use these ones, the ones you see in the photo here, which are compound microscopes, and I also use the scanning electron microscope. Okay, so this is how I get the plants ready to be seen under the scope. So after taking the samples, I place these samples in these 6 wells plates, and then I add the solutions of acetic acid and ethanol and glycerol, and it takes almost 2 days, and we need to change the solutions twice. So at the end of 2 days, the leaf tissues are like this, colorless. That's when I treat these samples with Trypan blue, keep them 1 more day, and after that, plants are ready, tissues are ready to be seen under the scope. And this is kind of what you see under the scope. This, the Conidia is going to stain blue, dark blue. And this process takes like 3 days. Now for the microcycle conidation experiments, what I did is I took leaves, cotton leaves. I surface sterilized the cotton leaves. Then I placed 100 microliters of spore suspension for Bavaria bassiana and 1 more set for Phyallimonium. I cover the leaves are placed on agar plates, not for culturing anything, only to keep the humidity. Then they are transferred to an incubator, kept at 27 Celsius for 5 days. After that, remove the leaf, put it in a centrifuge tube with Triton solutions, vortex for 2 minutes, and then plate on PDA plates. 3 days later, I count the number of colony forming units. Now with the results. By the way, this is a photo of Bavaria bassiana going into microcycle conidiation surrounding this stoma. You see the formation of new spores. Here, so I took a lot of the photos using the Trypan blue stain technique, but I'm showing you the ones that I took with the electron microscope because they're way better and more with way more detail. So in this case, the result. So I didn't see any spore germination on the leaves from my samples taken at 1, 3, and 7 days after the treatment was done. And maybe it's because the conditions in the growing room were not really conducive for these spores to germinate. But even when I didn't see germination, I'm not gonna claim that 100% of these things didn't germinate. It's possible that percentages of these spores, like a really low percentages, I would say in the 1 single digits, probably germinated, but I just couldn't detect it. So most of the time that when I saw these plants, these samples, I saw the spores, but not germinated. In this case here, I see the spores of Bavaria bassiana. One stoma, the length of this stoma is about 30 microns. And this spore here is like 4 microns. Same with Phyallimonium. I didn't see Phyallimonium germinating under the conditions that I grew my plants in my chapter 2. As I mentioned before, these spores, they have like a lemon shape. Didn't see this germinating. And also same with Chaetomium globosum. Never saw this Chaetomium germinating on the surface of the leaves. But then I was thinking, is it any way to make these things to germinate? That's when I did this. So I enrich the spore suspension with a and make a final 0.5% molasses and then I increase the humidity of this environment. Just what I did is I put the plants in a tray, and then the whole tray in a large bag for 3 days. Then after 3 days, I pulled the plants, I took the samples, and voila, these spores are germinating. Now we're talking. And I noticed germination for Bavaria bassiana, germination for inflatum, and germination of Chetomium globosum. I said, oh, this is great. And now, when these spores have germinated, I finally was able to detect some of these actually colonize the plant tissue, the leaf tissue in this case, through stomata openings. And you see here this the germination tube going inside the stomata, same with this Philemonium inflatum, and globosum going again inside this stomata opening. So that's when I finally realized, yes, colonization is possible only when you provide with the right conditions. And then since they germinate, let's go and see what happens with the microcycle conidiation of these two species, bassiana and Philemonium inflatum. So we have the two ones here that are phytogen plants, and these are or leaves, I meant, and these are delta pines. And you see the two species of fungi. You see the only Bavaria bassiana, which is the blue one, reproduces after 5 days of being incubated. You have significantly more spores in the first trial and the repeat, but nothing with Philemonium inflatum. And this experiment was done using only water. I didn't enrich the spore suspension with molasses. I just wanted to see what happened when you just do high humidity in water. And yeah, so only Bavaria bassiana was able to reproduce significantly. Conclusions for this short chapter. Fungal spores did not germinate in cotton leaves under the conditions I grew plants in chapter 2. Fungal spore germination occurred with the addition of an extra carbon source, in this case molasses, and high humidity. And plant colonization through stomata was observed. Bavaria bassiana reproduced through microcycle conidiation on the surface of cotton leaves and produced significantly more conidia or spores after 5 days of incubation. Now the take-home message. What is that I learned from this thing, from this experience? Well, I learned that mom recognition is more likely to be the mechanism involved in the response of phytogen plants pre-treated with dead spores. And to my knowledge, this is the first time that dead spores as foliar sprays are reported as potential plant defense solicitors. One size does not fit all when it comes to plant response to defense solicitors. And we saw that these two genotypes behave completely different. One of them recognizes the mom and the other one doesn't. And finally, growing, and I learned this thing the hard way, growing insect pathogen damage-free plants is key to detect differences in plant response to mumps and elicitors. With that, if that is not enough to get a master's degree, I did this work, and it was published a few months ago, and it was called Resin Cast Impressions as a Tool for Microscopic Observations of Fungal Epiphytes on Leaves. I did this because when I did this microscopy work, I noticed that because of all the washes that we do to get, the green removed from the tissues, probably is messing with, the original position of the spores on the leaf. It takes like 3 days, as I said, and it's expensive because you need several chemicals to do it. So I said, must be a better way to do it. And this was a very simple way to do it. And I was able to, with this resin, I was able to detect, like in this case here, these two, the first two are Boveria bassiana spores germinating. You see here the germination tubes. And also I was able to detect the Boveria bassiana going into a microcycle conidiation. You see the original spore here, germination tube, and new spores starting to be formed. Also saw Phyllalemonium inflatum, its reproductive structure here. And this thing is really difficult to get it with the trypan stain method that I did because of the washes. Because of the washes, you're not gonna see this structure here. And also you can just use it to see stomata and other features on the leaf. With that, I would like to thank Dr. Gregory Soar and the Charles R. Parencia Chair for financial support. Special thanks to my friend Cody Gell, Lauren Rivera Vega, Ashley Tesno, Elise Woodruff, of course, and Lauren Mayers, and to all of the current and former lab members. Thank you very much. And with that, I will take any questions.
All right, we'll give Cesar a round of applause. Good job, Cesar. Woohoo! All right, the way that we will do this is we'll open it up for questions from the audience, and then once we're done with that, we'll have Cesar meet privately with his committee, who will ask him some follow-up questions. So does anybody have any questions for Cesar?
No questions, good.
No questions. Good.
Can I have a question, Cesar?
Me too, Greg. So be after Julissa.
Who is first? You, Julio?
You go, Julissa.
I go? Okay. It's just a quick question.
Sure.
What is the difference between chitosan and chitin? Because in my case, I know more about it because I teach biochem and I focus a lot about the biochemical structure of these compounds. You can see difference very, very, very different, right?
Yeah, so chitosan is like a more complex molecule. It's like, I think it's an ester of
no, it's a polysaccharide.
Yes, it is. Yes, but it's in terms of inducing plant responses, it's being used like a chitin source, like a source of chitin. Yeah, that's what
you mentioned, like elicitor, right? For plant pathology classes, we use elicitors, and actually we use chitin.
Chitin, yes.
Chitin.
There are many experiments where they use either chitin or chitosan. Yeah. Yeah.
But it's a small difference, right? Chitin, I remember, is a polysaccharide with it's a modification of the sugar with nitrogen and an acid. And with the chitosan, you eliminate the part of the acid. It's still only, it's amino sugar.
I think so,
and
yes.
for biochem, yeah. Is, for example, the degradation product of chitin. Chitosan is a degradation product of chitin. And for example, in plant pathology, we use, for example, for the fungi, some enzymes are degrading the cell wall of the fungi. They are degrading chitin. So they are releasing elicitors. Those elicitors are mainly chitosan. And it's how the plant responds against them.
Yeah. Yes. Yeah. So we wanted to, yes, the closest thing that I could get to chitin was like chitosan, yes. And as I said, it's been used in several studies to demonstrate this amount trigger immunity due to chitin.
So thank you.
And Julio, I think you had a question.
Yes, I did, Cesar. Cesar, congratulations! Wow, what a great job you did! Tons and tons of work, and really good work at that. Plus, Cesar, so in the first experiment, where you had the viable and the heat-treated spores,
Yes.
was it me, or did you analyze the two spore conditions separately? Because towards the end, you concluded that there was this dead spore effect, and I'm trying to reconcile this two things. So were they analyzed separately, the two spore statuses?
Yeah, so I always did the experiment using both types of spores. For each genotype, I always included dead spores against viable spores. Yes.
Yeah. So have you tried putting them into your model, into your analysis for interactions, and to actually extricate that effect?
No, I don't think I have tried, because what I did is I combined the genotype data, the plants, the data from the genotypes by spore status. No, I haven't combined the two dead and viable spores in the analysis.
Okay, yeah, so I think it might be interesting if you did, just to see if spore status or viability is interacting with your other variables, genotype, I guess, and fungal species, right?
Yeah. No, I did.
Fabulous job, Cesar.
Thank you, Julio.
All right.
I have a question too.
And Carlos.
Cesar, and you may have mentioned, but I didn't get it. And obviously, you're getting a degree in entomology, not in plant breeding and cotton breeding. But do you know anything, the difference between these two genotypes in terms of any properties that would explain the difference between the genotypes and what you saw?
Yes. Good question. So one of these genotypes, phytogen 367, is a genotype of plant that is known to be tolerant to nematodes. And I know the nematodes, the other one, so both of them are GMO. Both of them have the Bolgar2 and the Roundup Ready genes. But the difference is phytogen, on top of that, is tolerant to nematodes. And I think that makes the difference. And I've been reading about trying to find why. It could be because it seems like this genotype is pretty sensitive to this mump, to chitin. And I mentioned that the fungal cell walls, one of the chitin is one of the components of this thing. But also I found that nematodes, they also have chitin. It might be that these plants are resistant or tolerant to nematodes because they are able to detect the chitin from these nematodes when they get in touch with the roots. That's why they are more hypersensitive to a mump recognition than the other one, than the delta pine. That's the best explanation I have of why one of these genotypes is responding to the dead spores or to chitin.
Yeah, you mentioned the nematode tolerance, and I figured they might have something to do with it. But great that you follow up on that one. I think Greg and Cesar finding out, that part of why some genotypes are being elicited by microbes in a different way. I think that's an area where industry is going to need a lot of help, because it's not, if you think about just the very few difference between the cotton varieties, or relatively, compared to the number of crops that have a lot more diversity. But anyway, I think that's an area to pursue, Greg. That's exciting.
Thanks. Yeah, the genotype's clearly been overlooked at a lot of people's peril. So we're Cesar's got some really good illustrations of that.
Cool. Thank you. Great job.
Thank you.
All right. Last chance for any questions. If not, we'll go ahead and thank Cesar again. Give him a round of applause, either in person or with your little applause emojis.
Congratulations, Cesar.
Thank you.
Very happy for you.
Thanks,
Great job.
All right, and thanks again to everybody from around the world tuning in. We've had 3 continents, at least 3 continents represented here for Cesar's defense. So thank you very much to everyone. And that just illustrates how important Cesar has been to people during his time here around the world. So thank you very much, Cesar.
Thank you, Consul.
All right, we'll have everybody else sign out. I think the committee will take, maybe if anybody needs to get up,
Break.
little break, and maybe we'll come back and start again in 4 or 5 minutes. Sound good?
Okay, is open for everybody?
No.
No, well, I mean, you can say hi and stuff right now, but the committee meeting will just be Cesar and the committee in a few minutes. But it's good to see you, Elisa. See you.
Nice
I haven't
to see
seen your
you.
face in a long time.
Yeah. Okay, good luck, Cesar.
Thank you. Thank you, Felisa.
All the best. You're going to do great. You will see.
Yep.
Have a nice Friday and celebrate safely.
You too. Bye.
Bye-bye. And we have Dr. Men tuning in from China. How you doing, Dr. Men?
Dr. Man.
Yeah, Dr. Men's on here. All right, well, I'll be right back.
All right. Ahora sí, tienen que cerrar la sesión ahora porque la siguiente parte es solo privada, solo con los miembros del jurado. Gracias por estar aquí, muchachos, familia y amigos.
Que te vaya muy bien, César.
Gracias, Julissa.
Nos vemos. Chao, chao.